Yeah.
Thank you. So I might be a bit the wild card today. I usually am. Talk about organ on chips, and I want to don't specifically go into details what I do myself, but actually more on what the field is doing and where can it go, where can it potentially help, or where can we connect, especially with the in silico part. And so our motivation in the lab is mostly to get patient stratification, or sort of the patient on the chip, by we essentially abstract the patient into a tissue model. This is in our case typically spheroids, but we also work with transval models or advanced 3D cell culture, and we then integrate them into microphysiological systems where we can connect the two different organs, or three, or also only one if you want, to get sort of a physiological environment, and use then these data to do an in vitro and vivo translation. That's where mostly you come in, ideally, but also, which is my very important statement for that field. We have to not only abstract it, but also turn back and get, again, back to the patient by doing this in vitro and vivo translation. So what do I understand by a microphysiological system? This is how you could see the schematic view, if you will. And I like to break it apart into three essential components. For one, it's the organ model, which is typically an in vitro model. Then we have the fluidic system around it and some readouts. The readouts can be sensors. It can be just collection of supernatant. Ideally, you have something integrated that is label-free. This would be the ideal, that you can read out distinct parameters of each individual organ model, if you will, and have parameters for each and every organ model you have in there. There are several concepts around, ranging from Harvard, the Wyss Institute, to MIT underneath, and also from other labs. And Berlin is one tissues system that can connect one or multiple organs in a microfluidic environment. They offer very promising solutions, but they typically still have some problems. They usually have tubings and pumps, PDMS and material that takes up everything that is in the cell culture, complex setups and complicated cell models. So I went forward and sort of thought a bit, what is the next generation microphysiological systems and what's are important? So our field has to at the moment show that we can validate or use this technology. We have to address the unspecific binding, as I said before, with the PDMS. We have to show key functions of organs which are represented in vitro. And they also have to be defined. What is a liver? What is a good model for an organ? And this is still to decide, I would say. And also the stability of the models over time in such systems. Important is also the aspect that I think the gain in predictivity has a price, and you always have to think about what is the real added value, because there are nice systems around where you can do very complex things, but maybe it's not even necessary. So why do the effort? Another problem is sourcing of cells and validating their biological reference. We start in tox, I mean, Hep G2s, they are a nice screening system, but they are not very good to model metabolism. So you need something a bit better, like HepRGs or even primaries. So what do we use, and how representative are these cells? And general challenges of in vitro also remain within these settings. So concentration in vitro versus clinical, drug-related factors, acute effects, rare clinical events, these kind of effects, they will also stay in these more complex microphysiological systems. So where can we use these systems? So I think there are two aspects that where we can use them. So on one side, it's safety, to look at key targets of toxicity like liver. You get using advanced cell models and flow, you typically get long survivability of the liver. So we can look at low clearing drugs, for example, kidney, cardiac, you can do test barriers, you can do efficacy studies by having better model system of the disease. But I think the real advance is by combining the two. So incorporating immune components or connecting organ models. And I think this is sort of the identification of translational biomarkers, model efficacy versus safety, and things like, for example, a therapeutic index, so that you can model in one system a tumor and the liver together, and then look at on one side efficacy, where you can see how efficacious is the drug on the tumor and how toxic is it, and at the same time measure both aspects, and you would get sort of the therapeutic window or therapeutic index within one assay. These are things that are sort of at the in reach, I would say, to do. So these assays are more aiming for replacing animal studies, not increasing the supportive tests. I put that in hyphens because replacing animals will most probably not happen, but it should add something where we don't have anything yet. So adding more filters for something that's already established is not improving anything. So we have to use these MPAs for integrated pharmacology and toxicology assessments, in my opinion, by connecting, for example, gut, liver, and kidney. This would add something that you can do typically in animals, but you could do it in vitro. And you have to focus on an area where there's a lack of tools, like the cross-reactivity of species. As soon as you involve the immune system, it's very difficult to do it with animals. Look at drug combination regimens or personalized safety. So, and why do I say that? It's mainly because there's a big shift in pharma portfolio. Up to about 2013, we were mostly dealing with small molecules where the pharmacology is, I'm not saying simple, but simpler. The question are organ toxicity, metabolites, biomarkers can go in vivo, and the approach is typically with animals and high throughput screening. And now we're shifting to something much more complex with drug conjugates, with peptides, with different antibodies, combination approaches, and this makes pharmacology much more difficult because it needs to involve a lot more key parameters. You need to model more complex systems, and the questions are also more complex, from cross-reactivity versus off-target to immunosuppressant and so forth. So maybe there's a real need for new tools, or not only maybe, I think there's a need for new tools, and I think MPS could be one of those. But that leaves me also sort of to the aspect of validation, which I think is important. There are a lot of interesting systems around there, and everyone talks how big and good and awesome they are. So I think one of the big problems at the moment is comparing them. So what data do we have? What do we get? How do we compare? There's a nice review in Advanced Healthcare Materials where they compare the different systems. They looked here at, you're not supposed to be able to read that, but they compared here liquid to cell volume ratios and metabolic burden between human body and in vitro systems. So what tool do we use in order to scale the tissue models, but also, how do we get the data and what do we do with it? Can we just take it one-to-one, or are we comparing apple with peaches? These kind of questions still are out there, and there's no clear standard on what is what. There's an initiative, especially in the US at the moment, to create these tissue chip testing centers where they try to test and characterize and promote the adoption of the technology. And they do the approach with in vivo information, in vitro information, and tissue chip results. They put that into a PBPK model and go to human clinical outcomes. This is the experimental and computational approach they use. And we're getting ready to send also our chips there and sort of see how well our chips are performing. They use platforms, cell culture medium biology to validate these systems. And this is sort of the testing strategy. They start with platform testing and drug plasma protein binding, go then to the biological NPS, and try then the pharmacological testing of the cokinetics, toxicology, and efficacy. And so I hope we can contribute to that they are actually starting to be adopted. And back to my motivation, I think it's up to us at the moment to generate and publish use cases of these systems. And I think especially this last part here of the in vitro in vivo translation is very important and needs to be added, because in vitro you can show anything if you don't compare it to something relevant. And with that, this is our team at ETH in the group of Andreas Hillemann. And we're working with Roche and Novartis and have quite good interaction with Pharma. It's quite an interesting field at the moment. And yes, that's it.